We need biodiversity to survive. But now, over 1 million species are threatened with extinction. And threats to biodiversity result in crises like new diseases, invasive species, and inconsistent food production. Major research programs use molecular methods such as DNA barcoding to track biodiversity on a planetary scale. Together, we can build a baseline of biodiversity data so we can measure changes due to climate change and anthropogenic activities and protect our biodiversity. This video presents a simple DNA barcoding 12-step workflow that can be performed with basic laboratory infrastructure. The easiest way to get started with DNA barcoding is to use commercial kits, at least for the DNA extraction step. Whatever kit's being used, make sure to follow the instructions. This is the equipment you'll need to DNA barcode using a tube-based method. Step 1. First, prepare all the necessary equipment and reagents following the protocol you've chosen. Read the protocol carefully. Step 2. Take a leg of the bug and place it in an empty labeled tube. Once you've taken tissue from the sample, change your gloves to avoid contamination in the next step. Step 3. Next, you'll add lysis buffer and proteinase K to break the cell membrane and to inactivate proteins such as nuclei that might degrade the DNA during this process. Take your pipette. Add a new pipette tip and then add lysis buffer to your DNA tube sample, our bug's leg. Discard the used pipette tip in the waste bin and return the pipette. Replace the cap on the lysis buffer and return it to its place. Select a pipette. Now you'll add proteinase K to your sample. Discard the pipette tip. Give it a quick spin in the centrifuge to collect all the reagents from the tube walls and the lid to the bottom. Step 4. The tubes are ready to go into the incubator or water bath at the temperature and duration of the time specified in the protocol. At this point, change to a new pair of gloves to avoid contamination. Once the incubation time is over, the tubes are ready for another spin in the centrifuge. Quickly spin the tubes again. The tissue lysis step is complete. Step 5. And now it's time to set up for the DNA extraction. You'll want to get 2 empty tubes and label them. Select your pipette, add a new tip, and then add your first reagent, your binding mixture. What we're doing here is we're grabbing the DNA from our original sample, in this case the bug's leg, and then running it through a membrane filter. Now we'll spin our tubes again in the centrifuge. Now we need collection tubes. Pop off the top of your tube with the filter attached and move it over into your collection tube. Then we'll wash our sample using the wash buffer in our kit. We're adding the reagents that get rid of the proteins and the salts, so in the end we're left with pure DNA. Once you've added the wash buffer, put the tubes back in the centrifuge. We'll move our sample to a new collection tube again and repeat the wash process to remove any residual salts attached to the DNA. Now we'll take 2 clean collection tubes and move your filters to them. At this point, we'll let them air dry. This will help remove the ethanol that's in the wash buffer. Next, we'll add our elution buffer and let it absorb into the membrane. When we do our final spin, the DNA is going to move down off the membrane into our tube. Now, we can discard the membrane and transfer the extracted DNA into our collection tubes. Now that you've extracted the DNA, you can take it to PCR. Step 6. Prepare all the reagents ahead of time. First, prepare the PCR mix. This can be pre-made and stored in the freezer. Second, organize the consumables and everything else needed. Label the PCR tubes. Step 7. Dispense 23 microliters of the mix into each tube corresponding to the number of samples, plus the negative control for DNA extraction, plus a negative control for PCR. This is to see if contamination has occurred during the setup of the reaction. Then add 2 microliters of DNA to the first tube, and 2 microliters of extraction negative to the second. To the third tube, the PCR negative control, add 2 microliters of water. Close the cap of each tube after adding the 2 microliters. Make sure all the tubes are labeled. Give them a quick spin to collect all the mix to the bottom of the tube. Step 8. Then, place the tubes into the PCR machine, or thermocycler. Close the lid and start the program needed to amplify the barcode region. Step 9. Before sending the PCR products for sequencing, it's a good idea to check if the PCR was successful. This quality check can be done through what's called a gel electrophoresis using an agarose gel. And here's how we do that. Take your agarose powder and add your buffer to it. Swirl to mix thoroughly. Then we'll microwave it and make our gels. Pour melted agarose solution into the gel tray. Then transfer the cast gel once it has solidified. Finally, place the gel into the TAE buffer-filled electrophoresis chamber. Step 10. Combine the dye with the samples and load the mix on the gel. Then, you top it off with a buffer and run it through a current by turning on the voltage power supply. Once the migration is done, the gel and its bands can be seen under UV light, and the image taken can be saved or printed. Step 11. Since we will be using Sanger sequencing, a highly accurate technique for reading a DNA sequence base by base, it's important to clean up your reaction mixtures so salts, primers, primer-dimers, and DNTPS are removed, and DNA fragments are selectively bound to the magnetic bead particles. Step 12. If everything looks good, the PCR products can be sent to a sequencing provider. The company will send back chromatograms or trace files in a specific format called ABI files. They need to be edited with special software, and once the sequence is finalized, it can be compared against a database of millions of sequences to find out what sample it was. Let's compare this sequence against the Barcode of Life Data System, or BOLD, to find the matching species. It's a Japanese beetle. BOLD is a searchable repository for barcode records. It stores specimen data and images, as well as DNA sequences and trace files. We've shown you a basic tube-based step-by-step method for DNA barcoding. With rapid advances in DNA barcoding technologies, many developed here at the Centre for Biodiversity Genomics in Canada, we are tracking life at an unprecedented speed. Automation and high-throughput sequencing technologies have scaled up DNA barcoding to enable species discovery, exploration of species interactions, and their dynamics in a way that has revolutionized our understanding of life.